is a hormone that's produced by bone, specifically these cells called osteoblasts, and it has major metabolic effects throughout the body, including on the pancreatic beta cells, improving their ability to produce insulin, the Leydig cells of the testes, and on the brain. So these osteoblast cells within their nucleus have a gene called BGLAP, or bone gamma carboxyglutamate protein. So this gene right here is regulated by a milieu of transcription factors, and it eventually codes for a protein called osteocalcin, but this protein is called GLU osteocalcin. And this protein undergoes a post-translational modification into something called GLA osteocalcin. And this osteocalcin is a 49 amino acid protein, and three of those amino acids out of the 49 are glutamate residues. So they're glutamate. And what happens is this cell will modify those three glutamate residues to form a gamma carboxyl glutamate residue. And what that means effectively is it's doing this reaction right here, where it's taking a glutamate residue right here. This is our little glutamate residue right here. And it's adding a carboxyl group to that glutamate residue. So I have two carboxyl groups bound to the same carbon, and each carboxyl group has a negative charge on the oxygen, which allows these two negative charges to coordinate or chelate with calcium. So they're able to bind with calcium, Ca++, because calcium has that +2 charge. And so that negative charge is able to coordinate with calcium. And so functionally what we're doing is we're taking a protein, we are modifying it into a protein that can now bind with calcium, and calcium is ever present in the extracellular matrix of bone tissue. So I'll go back up here. What's happening is we're transcribing this gene into mRNA and translating it into a protein, osteocalcin, that contains glutamate residues, and then using this enzyme called GGCX, or gamma glutamyl carboxylase, I'm converting this glutamate osteocalcin, or GLU osteocalcin, into a GLA osteocalcin. This is the storage form of osteocalcin because it has that gamma carboxyglutamate residue that can bind with calcium. So I'm storing it in bone. And this gamma glutamyl carboxylase is vitamin K dependent. And so it's using vitamin K, and what it's doing is it's turning vitamin K in a hydroxyquinone form and converting it into vitamin K epoxide. In the process, it's able to add a carboxyl group to those glutamate residues. And usually the vitamin K that's dependent in this reaction is vitamin K2 in either the MK4 or MK7 form, because there's different tissue distributions of vitamin K. Vitamin K1 tends to be in the liver and involved with the coagulation cascade, whereas vitamin K2 is present in bone tissue, or it's partitioned to bone tissue, so that this enzyme can use it in this reaction. So this is a vitamin K2 dependent reaction. And so I'm converting vitamin K in its hydroxyquinone form, or the reduced form, into an epoxide form. And in order to recycle that vitamin K from the epoxide form back into the reduced form, I need this enzyme to be present, called VKOR1, or vitamin K epoxide reductase 1. But once I form this GLA osteocalcin, it's then put in these little secretory vesicles, and it's exocytosed out of the cell, where it binds with calcium located on the outside of the cell due to those two negative charges coordinating with the two positive charges of calcium. And calcium is present in hydroxyapatite. And then when I'm reabsorbing bone, or when I activate an osteoclast, so these cells that start breaking down bone, osteoclasts will start decreasing the pH or increasing the acidity of the extracellular matrix of bone. So what they do is they start pumping hydrogen ions to the outside of the cell, where I have a lot of hydroxyapatite and calcium that has been sequestered. And that allows this osteocalcin that has been gamma carboxylated, that's bound to calcium, to now unbind, because what's happening is these little carboxyl groups, these little O minuses, are being pronated by these hydrogen ions that are being pumped out by this osteoclast. So you can see here, over here I have OH and OH. So it's no longer bound to calcium, which allows this osteocalcin that was previously stored within the extracellular matrix to then diffuse into circulation. And once it's in circulation, it's now in the activated form of osteocalcin, or the uncarboxylated form of osteocalcin, which can then have major metabolic effects on a broad array of tissues. And so this is the general mechanism by how osteocalcin is produced and released into circulation.